Professor Rohit Dutt, Dean, GT Goenka University, Gurugram. Dr. Rohit Dutt is presently working as in School of Medical and Allied Sciences, GT Goenka University. He is instrumental in developing new policies to improve academic structure at GT Goenka University, keeping in view of current need of various sectors. Professionally, he is a medicinal chemist with expertise in computer-aided drug design. He has done PhD in pharmaceutical sciences from MD University Rohtak and M Pharma in pharma chemistry from Guru Jamshedwar University Hisar. He is an alumnus of STG RITS Dehradun at graduate level. He has 16 plus experience of teaching experience. His expertise in the analytical method development and validation, natural product chemistry, computer aided drug design field enable him to conduct research in area of diverse nature. His research area include lead identification and optimization, chemical computation. He has more than 60 research and review publication, most of them in international journals of reputed to his credit, in addition to 3 book chapters with international publisher. He has been involved in 2 consultancy projects, namely Tuning India and Rishi by University of Deusto, Spain, funded by the European Union Erasmus program. He is a certified auditor for a accreditation of labs by NABL. He had also honored with APTI Best Young Pharmacy Teacher of the Year Award in 2015 and Nirali Prakashan Award in 2019 and the Teaching Excellence Award 2021 by ASMA for his excellent contribution in the field of pharmacy education. Dr. Dutt has been awarded with the excellent contribution in the field of the pharmacy, the Teaching Excellence Award 2021 by ASMA for his excellent contribution in the field of the pharmacy education. Dr. Dutt has been awarded with the prestigious silver medal by His Excellency Honorable Governor of Haryana in recognition to his PhD research work. He has delivered more than 100 guest talks on various topics of healthcare domain and organized healthcare camps in rural area in Delhi NCR. I welcome you, sir. Sir, I please to address the participants. Thank you so much.
Thank you, Dr. Prerna, for your generous introduction.
Thank you, sir.
And good morning, one and all who have joined on a Sunday, despite a holiday. You are devoting your time for some knowledge exchange. I think it's good for the profession. And I congratulate the organizer of this AICT program, because Guru Gobind Singh College of Pharmacy has always in the forefront in the state of Haryana, the first college who has been accredited by the NBA. And I think continuation of the NBA is the real validation of all the good practices happening at Guru Gobind Singh College of Pharmacy. And I'm very much close to this institution, and I learned so many things at the platform of Guru Gobind Singh College of Pharmacy. So it's always good to interact, good to contribute back in any cause which is organized by Guru Gobind Singh College of Pharmacy. So once again, I thank the organizers and congratulate the entire team of GGS COP for organizing such events time and again, which gives a platform for the various experts in different domains to express their views, to express their knowledge. So I have taken a simple topic today, since the theme was related to some experimental research to plant something in a particular domain. So I choose a topic which is of general interest. May I share my screen if you can allow?
Yes, sir. You can.
Is the screen is visible, Rohit?
Yes, sir. It's visible, sir.
So, thank you. So I have taken a simple topic of development and validation of an analytical method. And if we talk about an analytical method vary from the titration, the traditional titration methods to, you can say, HPLC, MSMS these days, and UPLC these days. So there are wide variety of analytical methods which can be validated and which can be developed. And since I cannot cover each and every topic in a lecture of 30 or 40 minutes. So I restrict my talk to LC method, that is the liquid chromatography. And most of the, why I have chosen this technique, the reason is obvious that most of the industry are now working with the LC technique, and 90% analysis is done on LC method, whether you do dissolution or whether you do any assay. So it's a ongoing work of 24 hours. So it's always better to have some idea. I am expecting that the faculty members have joined for this program, and they have the basic understanding of liquid chromatography method. They must be teaching also. They must have gone during their M.Pharm or B.Pharm studies also. So I'm not touching the basic concepts of liquid chromatography. A brief introduction, and I will directly come to the planning strategy for a development of a LC method and how to validate this LC method. That is more important, I think. Then you can always deliberate. You can come in the end with your questions also, so you can put your questions in the chat box. Rohit, please confirm whether the slides are moving.
Yes, sir. It's moving, sir.
Yeah, thank you. So I think everybody is aware of a famous scientist, or scientist, if we specify the domain, a botanist. It's a Russian botanist, Mark Tessler, who is the first person, and I will say in terms of regulatory that he is the innovator to establish the principle of chromatography. What he did in 1906, he presented a paper in 1906 where he described how he filled a glass tube with a chalk powder. Chalk powder means calcium carbonate, and allowing an ether solution of the chlorophyll, which is active component, to flow through the chalk powder. And thus he separated the chlorophyll into layers of different colors, and he then called this technique as the chromatographic technique. So I think everybody is aware of this innovator, and it's always better to recognize the efforts done by the innovator rather than going straight into the techniques. So chromatography, in which mobile phase is a liquid and stationary phase is a solid. Everybody is aware about this thing. Just a minute, I need to just settle down. Thank you. So liquid phase, liquid is used as a mobile phase, which is called as diluent, and the stationary phase is a solid, which can be a silica particle. These days, Kieselguhr is also used, and there is various alteration has been done to the normal silica particles to achieve the maximum separations. So it ranges from the type of material. The changes are in terms of the particle size of the silica particles. So depending upon the nature of the sample, there are different columns, different stationary phase available in the market. So the basic concept will remain the same, whether the technique is of high end like UPLC, but there will be a mobile phase and there will be a stationary phase. And stationary phase is responsible for the retention of the most of the analyte molecules, and mobile phase will temporarily involve in the hydrogen bonding or the interaction with the analyte molecules. So yes. So HPLC is the extended you can say, form of liquid chromatography, which is generally used these days to separate compounds of various interest. So it's a packed instrument. Everybody knows about that. And you have seen the HPLC, the component of HPLC, that it contains a reservoir for mobile phase, a pump to deliver the desired mobile phase to the column, and an injector to inject the sample. And injectors are also these days, they are manual. They can be manual also. They can be automatic sampler also. Automatic sampler work for 24 hours without, you just made a program in the HPLC, and the entire HPLC will run for entire 18-24 hours without any human intervention, and you can collect the chromatograph in the morning. This is the way how industries are running these days. That's why this HPLC is very important for the point of student placements also. If a student is well versed with the concept of HPLC and he is comfortable in running HPLC, then I think he can definitely get a job in the quality control or quality assurance department, because it's a 24-hour job. So shifts are changing, but HPLC is running the sample day and night. So compounds are generally separated when we inject sample mixture into the column, and on the basis of different detention with the stationary phase, compound generally separates out, and it is the normal partition behavior which we have studied in the by the term of chromatography. There are different troubleshooting can be done to prevent your chromatograph, to prevent your analysis. Like the first thing you need to understand the pressure. So pressure need to be controlled. It should be below a certain limit. It depends upon the type of HPLC you have. And this is the one limitation before development of any method. So your nature of sample and the pressure, your pressure of your HPLC, available HPLC. I'm not talking generally that every institute have the high-end HPLC. They may have the basic version of HPLC, which can only tolerate a pressure of 4000 PSI. So you need to look into these parameters first. These are called instrumental parameters, which need to be undertaken before starting any project. The type of columns you have reverse phase column, you have normal phase columns, or you have ion exchange columns. So this is the limitation, I think, at the institute level. So if want to start a development program or want to enroll a student of M.Pharm or PhD for this development part, they must ensure the resources first, what kind of resources you have to start a development process. Because I'm restricting my talk to the resources itself, that I have seen that most of the institutions, the private institution, doesn't have much high-end HPLCs. So there are limitations. So I'm mapping the limitation within my talk itself. Otherwise, I can say that you can do the analysis with your PDA detector, and it costs very high. Then it's not possible for every institution to afford a PDA detector until and unless there is a grant from the government agency. So different types of chromatography. You have heard that what is normal phase, one is reverse phased, and third one is adsorption and ion chromatography, size exclusion chromatography. These are different phases of normal chromatography. And normal phase means normal, you haven't changed anything in the silica particles. So that's why the name is given normal phase. And when the normal phase silica particles are there, so obviously the polar analytes will interact and the non-polar will wash away quickly. So, but in organic chemistry, in pharmaceutical chemistry, you have seen that there are so many non-polar molecules. Most of the organic compounds are non-polar in nature. So scientists thought that how to separate these compounds from a polar phase. So they reverse the phase, means the nature of this normal silica particles, which is polar, they made it non-polar by end capping or end carbon loading. So they reacted the surface tylenols and with a non-polar chain of carbon, which is known as carbon loading. and the surface is now become non-polar. So now for non-polar kind of analytes, we can use this reverse phase chromatography, and 90% of chromatography these days are used in this this phase only. So I think you can target this phase. Reverse phase is the choice of interest for pharmaceutical industry these days. Although, so if the compound is ionic, you can always go for ion exchange chromatography. So there are different columns which are responsible for this ionic exchange separation. And if the molecular size is of the particles is big and more than 2000 Dalton, then you can always go for size exclusion chromatography. If your mixture have different analyte components which are varied in size, so you can always go for size exclusion chromatography. So these are the major domains, major separation forms of liquid chromatography, where you can look into your option, where your compound fits better. So you need to look into the properties of your compound, that whether the mixture contains polar compounds, whether the mixture contains a list of non-polar compounds, whether they both contain a mix of compounds. So depending upon that, we can use the column needed for a particular separation. So this is a typical diagram where you can see that computer control, and these days Empower software is used with Waters, and Agilent use different software. These two are, and Shimadzu is also coming up with some good HPLC, UPLC instruments. So this is a basic diagram of the HPLC, where one need to understand that whether you are able to have a knowledge of your computer software, because computer software is very much needed for integration purpose. So it's a part of development process. So you must know how to integrate a peak and what are the different features which are needed to fetch the system suitability parameters. So after running anything, you need to do the processing of the data, and that is very much important. And that is the task of a faculty colleague, that he or she can train the students on this software so that they can understand. The basic concept will remain the same in each and every technique, but one need to understand the basic criteria of a system suitability parameters. So these are in the center, you can see there are two pumps in this HPLC system, a manual injector where the injection port is written, and a column is attached after the injector port. An ultraviolet detector is there. So the mobile phase is flown through the entire system by the use of these reciprocating pumps, which use the back and forth pressure, and they fill the mobile phase and they transfer it to the column. So this is a basic technique I think you must have gone through in your studies and in your classes also. So we can internal diagram of this HPLC, you can just take a look in the internal diagram of this chromatograph, that we can use 4 mobile phase at a glance. 4 mobile phase are basically used for gradient elution. There are 2 isochromatic, one is isocratic, one is gradient elution. So whenever you are using more than 2 solvents, you always have 4 reservoirs with you. So it's a limitation. You must check with your HPLC system whether there is a provision of running 4 system simultaneously, or only you have the single pump or a double pump binary pump. So you before development of any method, you must look into this. And a simple example why I'm saying this is that if we are going for a development of a test for related substance means the impurities, and impurities can be many in a single drug. So suppose you say paracetamol has in process impurities of 7 impurities are listed in paracetamol by synthesizing. So if you are developing a method for checking the impurity level, you need to run the gradient elution. But if you are having only isocratic mode or a binary mode at your institution, I think it's difficult to develop this method at the institute level. You need to go for a gradient elution for the proper separation of multi-component analysis. So that's why I'm saying that in the resources available with the institution or with the scientists is of paramount importance before assigning any project to a candidate. So you can look into the pressure also, the pressure range of your HPLC. If the pressure range is less is 4,000, then I think you cannot do the samples of herbal extract or the plasma very well, because it may lead, in case of you are using buffer or you are using any biological samples, the pressure rises. So there is a risk of clogging also in the column. So you must look into that, whether we can use the plasma sample on the column or not. You need to demarcate whether this column is fit for the pharmaceutical drug analysis only in the samples, or you are going to use the column for the plasma. So there are different columns available for the different purposes. So you need to look into the resources, and you need to look into your injector system, whether you have manual injector system or you have automatic sampler. If you have automatic sampler, it's very easy to run the validation and other things. Otherwise, the scientists need to sit there and he need to manually feed the injection time and again when the run time is completed. So it's a tedious process. Going ahead with the topic or the theme of today's discussion, that why there is a need to develop a new method. It's always happened that when most of the pharmacopoeias generally publish the methods for most of the drugs in the market, when it is approved by the government, the pharmacopoeia develops these methods, and the methods are published in different pharmacopoeias and different compendiums also. So why there is a need to develop this method? So the first thing is that whether there are methods available in the literature. Yes, if the method is available, then why you are doing it? So you must have the clear idea, because these days you have seen so many methods developed for a single drug as a part of MPharm thesis or as a part of PhD project. Why you are doing it, I cannot understand. I have seen so many papers, the same mobile phase, the small variation in the mobile phase, small variation in the column length, and small variation in the this flow rate, and a new method is developed. This is not the purpose of new method. You must understand the severity of this thing. We are unnecessary, for the sake of doing project, we are doing this. Don't do this. If there is a genuine reason that there is no method available in the pharmacopoeia, or somebody has patented the method, so there may be some chance to develop a new method. Or you have seen that if you are doing some simultaneous estimation with some other drugs and the method is not available, so you can always go for a development of new method if your formulation is different, if your dose is different. Some genuine concern should be there, some genuine research question should be there before developing any method, rather than going straight, taking a drug and developing a new method. It will not suffice the purpose. So the purpose should be very, very much documented in terms of the genuineness. So this is the real limitation of today's research, but what I have seen in the literature. Or if the existing methods are there and they are producing so much of error, if you can highlight that, yeah, that is a point of research question, that you can go for a new method. And if the existing method is not providing the adequate sensitivity to a particular analysis, that maybe the method has been developed for assay and you want to do the content uniformity or the blend uniformity or dissolution, so, and you are not getting any published method, then you can always go for it. And pharmacopoeia generally do extensive exercise to develop method. And although I understand that some methods are very tedious, but what the industry generally do, industry look for the shorter runtime. They want to save the cost of the analysis also. So sometimes they develop the method on the basis of increasing the speed of the analysis in the industry, or cost-effective methods they wish to develop. So there must be a reason. The nutshell outcome of this slide is that there must be a strong research question why we wish to develop this method. Otherwise, developing a method and just playing with the parameters of SPLC, I think this is not the right way to develop a method or propose a method. I think it's a waste of time. We should concentrate on the real work. So what are the strategies which can be used to develop a method? I think everyday chromatographer develops new methods, and it is published at different ends. So we can look into the literature also, the available literature for a particular analytical method, and we can modify a bit as per the requirement. But there must be a research question that what is different in your analysis, what is different research question in your sample. So you can go for if a HPLC method is published in a good literature source, say suppose Journal of Pharmaceutical and Biomedical Analysis, or it is listed in Pharmacopoeia also, and you have a different composition of the final test concentration, or you have added some additional molecule, you can always do that. So one good strategy for a speedy method development is that if the literature is available, if the chromatographic parameters is available, you can always look into this. Okay, so it's not bad to look into literature. It's always good to ensure the literature. So sample permission and instrumental condition are adopted to make use of the latest method and instrumentation. Usually a compound with analytical method exists that is similar to sample of interest. So another way out is that we can look for some of the compounds of similar structure. The second strategy can be some of the compound with the similar chromophore or similar structure. I think for the start, for the development phase, we can look into the chromatographic parameters of those structures also. A series of, you can say, benzodiazepines, and there is a small variation in some of the benzodiazepines, and if method for X benzodiazepine is available, then you can take it as a base method for the development of method for the Y benzodiazepine. So this is another strategy. And the third strategy obviously is the traditional one. You need to go into the physico-chemical parameter first. And a book by Snyder, a very good book by Snyder. If anybody wants to develop a method, just go through a book of Snyder HPLC, practical HPLC method developments, and it's a Remington of HPLC analysis, and you will learn the content very precisely, how to develop, how to control the parameters, and how to develop a method at its own, rather than looking for literature source. These are the speedy methods which generally industry used, or these days the scientists use these methods. They don't have the time to devote much going back into the traditional method. Although pharmacopoeia, the IPC and the other pharmacopoeia persons, they generally scientists involved in the developing monographs generally do traditionally. So it's dependent on you which method you want to take. So what are the steps which are involved in the development of the method? So when developing an HPLC method, the first step is always to explore, as I told, the literature. And second step is then the information of the sample. And before beginning method development, one need to review the available information on the sample. For example, physical chemical number of compounds in the sample. How many compounds are there in your sample dosage form? Dosage form means any dosage form, tablet, capsule, any nanoparticles dosage form, whether it's a liquid dosage form or it's a solid dosage form, or it's simply injectables. So you need to understand the type of dosage forms as well as the number of compounds in the sample dosage form. One can identify and list out the possible related substance through a synthetic pathway of the drugs. You can look into these, the possible impurities must be there, the process impurities must be there. So you can look into the synthetic route of that particular drug, and you can list out the possible contaminants or possible impurities which may be available during the with the sample, and you can look into those impurities in your chromatogram also. So otherwise, every vendor who supply the drug, the API, they generally give a COA certificate, the certificate of analysis, which clearly lists the percentage of impurities available or percentage of impurities notified with a particular drug. You need to look into that, because that percentage of impurity must be in a certain limit over the period of time. That is the if you are going for a stability indicating analytical method, you must look into the level of impurity. Otherwise, these level of impurities rise over the period of time, and you need to ensure the shelf life of your API. Suppose a company has given you a gift sample and the shelf life is near to expiry. Generally, it happens whenever we ask a sample from the company, the reference sample, they generally provide the samples near to the expiry. I have seen in some cases, and you must ask for the certificate of analysis. Sorry, you must ask for a certificate of analysis. So that will give you a clear picture of the shelf life of the API, because near to the expiry, it may possible the level of impurities are much more than which is expected and which is acceptable. So please look into that, the shelf life of the API, rather than going straight away with the development part once you receive a active ingredient. So sample information provides important clues on the best choice of initial conditions for HPLC separation. Some chromatographers, they use theoretical approach on the basis of chromatographic profiles of similar structures, and other may use the empirical approach. Two approaches are there on the basis of sample information. So the sample information which you need to collect is molecular weight, pH and pKa value of the compound is very important. You know the relationship between pH and pKa. So basically the separation which are pH sensitive, you need to look into this factor also. The UV spectra of the compound is important, whether the compound has the presence of conjugation, a double bond or triple bond in their structure, whether they are single bonded compounds. So single bonded compounds are not easy to analyze in the UV spectroscopy. And similarly, we cannot use for HPLC with UV spectrophotometer. The most of the spectrophotometer which are available with the institutions, they have generally UV visible detector with them. So it's a kind of a UV visible spectrophotometer itself in the HPLC system, fitted in the HPLC system. So if the compound or the analyte is not UV sensitive, then obviously you cannot develop a method of that particular HPLC. So you need to look into other options, whether you are going for effective index or something, some other properties of the compound. So, but you need to ascertain that whether these resources are available at your end or not. So concentration range is another factor which is of interest, that in which concentration range you wish to work, and how to determine this concentration range. You need to calculate the final test concentration of your dosage form, at which concentration your sample is going to be analyzed for. Suppose for assay, if the final range is say 10 ppm, so you can work on that your methods should be sensitive at this range. So concentration range is very important. And if you go through the literature, if you found a drug where the concentration range is 100 ppm, so you need to look into more modifications, because 100 ppm, if the concentration range is 100 ppm, you need to look into the sensitivity at 10 ppm also. So this is important. You need to list out these parameters while you are doing it. Molecular weight, again, I told you that if the molecular weight is more than 2000 Dalton, I think it's a waste of time on HPLC technique. You need to look into the techniques. So solubility, again, it's a like dissolves like. Solubility is an important factor that in chromatography. So polar analytes attract with the polar columns, and the non-polar analytes attracts with the non-polar uh, non-polar compounds. So both columns are there, normal phase and reverse phase. So solubility is important. You must be have a clear idea that the compound is polar or non-polar, in which solvent it is soluble. So this diagram I had taken, it was a big diagram. I was not able to copy it very well. So you can look into that. If the sample weight is more than 2000, it's less than 2000 Dalton. The compound can be water soluble, compound can be organic solvent. So on the basis of different cationic, anionic phase and the polar and non-polar properties, you can select a variety of columns, that amino, cyano columns, the reverse RPC is the reverse phase column. So a BPC is the normal bonded phase column. So different column you can look into this chart. You can visualize, you can map your drug information, and you can look into which particular column is suitable for this particular analysis which you are looking for. So you can always look into this chart. You can have the idea to starting point, where to start, how to start with. So this is the diagram which will give you an idea. You can map it, the properties of your drug, and you can list out the probable columns which will work for your analysis. And these days, since I told you that so many transformation and the advancement has been done in liquid chromatography, design of experiments, analytical design of experiments is the term given for the prediction of these chromatographic parameters. So you can always use a software, and you can always use the analytical design of experimentation, which can give you a predictive analysis that the probable columns and other parameters which are needed for a development of a chromatographic separation. You can always look into that. So it's a recent technique, but I think that needed a separate 40 minutes discussion. So I will restrict it that you can go through some of the good videos, YouTube videos available on design, analytical design of experiments. And if you have the software, you can always predict the analysis. Unlike QSAR, we can always go into the prediction of the chromatographic parameters also. And so many papers have already been published on the prediction, and then they do the validation with the experimental setup. So it's a good exercise if you do a part of in silico, predict the chromatographic parameter, and then compare it in the real sense. So, so second step is the define separation goals. Separation goals means sorry. So recently I've been month back I've been tested COVID positive, since I'm feeling some of the side effects even after COVID, and now negative status. So the next is the separation goals. That what goals you need to set for your separation means you want to go for qualitative analysis, or you want to go for quantitative analysis, or it's a plasma sample, or you are going to develop a method for your herbal extract, or so something like that. So you need to define it, that which kind of sample, which kind of analysis, are you going for assay, are you going for dissolution, which kind of sample you are going to analyze, because sample comes in different forms, like ready to inject. So direct so direct injection of the sample is preferred in and convenience, and with a greater precision, if, like in parentals. However, most samples of the HPLC analysis require weighing and volumetric dilution before the injection. Best results are often obtained when the composition of the sample solvent is close to that of mobile phase. So that is the first point that you must look into when you are developing a mobile phase. So look into the mobile phase composition. The composition should ideally remain same when you are diluting it as a volumetric for volumetric dilution. If the mobile phase is you are making dilution with the mobile phase, I think it's a win-win situation. So best you will receive the best results when the composition of the same solvent is close to that of mobile phase, because it will minimize the baseline upset and other problems. And some samples they generally required some special procedures before analyzing directly. So some sample like partial separation or pre-treatment prior to HPLC because of need to remove interferences, or they may be too concentrated. We need to dilute the samples, or we can eliminate the column killers. I say that this is a column killers. Anything which is of which is not mapped to the pore size of your columns, which is hairline pore size, I think it's killing the column. You need to ensure that such concentration should be diluted, such concentrates should be diluted before reaching to column. Otherwise, it will affect the column and the column will be blocked. So it is important to always know the nature of the sample matrix and the probable concentration of various analytes. In many cases, the development of adequate sample pre-treatment procedure can be more challenging than achieving good HPLC separations. So the first task is, if there is a need to extract a sample, suppose in the tablet dosage form, you need to put a solvent where your analyte drug is dissolved and rest becomes insoluble. So you need to select the solvent. Sometimes you are successful in achieving a good separation, and sometimes it happens that interferences are there from the other bulk of the tablet dosage form or the capsule dosage form. Before the first sample is injected in during the HPLC method development, we must be reasonably sure that the detectors selected will sense all the components of interest. So it is of another interest that you must ensure that all the notified components they must be UV sensitive. Otherwise, it will be difficult that you will see that on the sensitive compounds will show the peak and non-sensitive compound peaks will not be there in your chromatograph. So variable wavelength ultraviolet detectors normally are the best, are the first choice because of their convenience and applicability for the most sample. For this reason, information on the UV spectra can be of important aid for the method development. The UV spectra, easily, if you have PD detector, you can scan it. If you have a UV detector at your end, you can scan it for the determination of UV maxima. Maxima can be 1 or 2 or 3, and you can select the maxima, the best maxima out of these three samples. We generally avoid maxima near to the UV range, starting range, that is 200 to 220. We generally do not look for analysis at 200, 210, 215, because most of the solvent shows their peaks or absorb strongly in this region of UV radiation. So you need to avoid these maxima which are close to or matching to the solvent. So you must ensure while selecting a solvent, the maxima of the solvent must be kept in mind before selecting the analyte maxima. So ideally, if you are working in a UV range of 200 to 400 nanometer, maxima near or above 250 or 240, and below 390, I think it's a good range to work for. After getting this sample information, I think you can look into the nature of sample. I have told you that the sample can be categorized. If it is a volatile sample, then you can always go for GC, you know, to go for liquid chromatography. So it's a basic chart. So sample can be of two types. One is regular sample and one is special sample. So after getting the sample information, you can look into You can categorize your sample as per whether it is a normal neutral sample or a ionic sample, or it's a special sample which is inorganic ions or isomers. Enantiomers means chiral chromatography will come into the picture. So you can look into this. So the regular samples are considered as those typical mixture of small molecules having a molecular weight less than 200. So this figure outlines the strategy which are recommended for choosing the experimental conditions for a first separation based on the knowledge of the sample composition and the goal for a separation. The first question is which chromatography will suit or promising for a particular sample. In this, HPLC has been, I think I say that HPLC has been chosen as a best chromatographic separation for a particular sample. So you can go into then isocratic or the gradient dilution. Isocratic means that if the components are not more in your sample, more than so you can say 3 to 4 samples are there, then you can always look for the gradient dilution for the better resolution of the peaks. Otherwise, you stick yourself to the isocratic one if the sample analytes are less in number. So this and the different this special on the special needs special treatments. So these ionic isomers, every biological samples like peptide, carbohydrate, nucleotides, they need the different columns and the different development procedure. So first you list out in which category the sample your sample fall in regular or in special. If it is regular, then which kind of technique you are going to use: normal phase, reverse phase, ion exchange chromatography, then mode of separation, isocratic or gradient, or you need to add some. If it is ionic in nature, you can need to add ion pairing reagent also. So characteristic of a primary HPLC method, you can look into that which chromatography is more beneficial to what kind of analysis. So the reverse phase usually use the water organic mobile phase. The reverse phase, the silica particles are non-polar in nature, so obviously the organic mobile phase is done through the column, and it will temporarily distort the interaction and the molecule will move and you will see the peaks coming on your chromatogram. So the basic. So it's a first choice for most samples, as I told you earlier, especially for neutral and non-ionized compounds that dissolves in water and other things. Similar polar solvents. Ion pair HPLC use water organic mobile phase. And, obviously, when we use the word ion, it need to control the pH. So for controlling the pH, you need to have a buffer. So there are the reverse bonded phase columns with cyano columns, and it is acceptable for ionic and ionizable compounds. Normal phase, as we are not changing any surface particles, we are keeping it normal. So polar phase, stationary phase is there, so non-polar mobile phase will be there. So columns generally comes cyanos, dialos, minus silica. It's a good choice. When reverse phase or both the techniques are not they are ineffective. So it is used for lipophilic samples that do not dissolve well in water and organic mixtures. So initial sample choice for special samples, you can say, as I told you, that if your sample is coming under the special sample that needs some treatment, so inorganic ions, so you can use ion chromatography. Isomers can be separated with some reverse phase column also, but better separations are generally achieved for isomers, especially with the reverse phase column with the cyclotesting silica column. So you can always look into these types of columns, and these types of columns are very exclusive, and generally R&D centers have these kind of a very wide variety of columns. So you need to look into the resources also, whether you are able to find these columns or not. And the number of compounds which fall in these categories are also not much. That's why generally most of the analyses are done on the reverse phase column. So in enantiomer, if you have enantiomers, obviously you need to produce a chiral conditions for their separations. And biological samples always need a special kind of treatment. So you need to look into the columns morphology and other factors of how to extract the analytes from the sample. And the macromolecules, obviously, the size exclusion chromatography is the better choice for the big molecules. So if we talk about how to go about, since you have listed out the properties of your sample and you have listed out that your sample is regular, I'm not talking about the complex samples, and you have listed out that some of the information was available on the mobile page in the literature, or you are not aware about the information on the mobile page. Just you can use the solvent in which the drug is soluble. So these are the starting conditions where you can start your initial run. It's a trial run just for the analysis of how to go about how a molecule behave on a particular chromatographic conditions. So you can look into this, that column. You can start with the column of 15 into 0.46 centimeter with a particle size of 5. These are the normal parameters, I think, which is available with each and every institution if they run the HPLC. So you can look into this. You can use two solvents. One can be buffer and one can be acetonitrile, or otherwise, if the drug is not ionic, you can always use the water acetonitrile combination. So you need to modify the percentage of B. B is the acetonitrile, the organic phase, the aqueous phase and the organic phase. So A stands for aqueous phase and B stands for the organic phase. So most of the samples, unless they are very complex, short columns are recommended to reduce the method development time. That's why I mentioned the short dimension, short column. Always look for a shortest column available with you. Ultimate outcome should be that we should do the analysis in a speedy manner, because we need to run the HPLC for 24 hours and number of sample is very much. So need to look into the runtime also very critically. That we must unnecessarily we are increasing the cost of the compound. Ultimately, the analysis cost will add to the cost of the product itself. So you need to look into the shortest runtime in which that sample eludes with the system suitability parameter, acceptable system suitability parameters. A flow rate of 1 to 1.5 ml per minute should be used initially, and packing size can be of 3 or 5 micrometer. So reverse phase obviously is a choice for many of the samples, but if the acidic or basic analytes are present, then reverse phase ion suppression can be used for weak acids or bases, and reverse phase ion pairing is used for strong acid and strong bases. The stationary phase should be C18 bonded to start with, and for low or medium polarity analytes, normal phase HPLC is a potential candidate. So if the separation of isomer is required, obviously the sign of bonded phases are easier. So we are not talking about those special samples. The sample size can be less than 25 microliter, because big sample size may result in the broadening of the peaks. The peak may be broad. So if you have again one more important parameter is there, thermostat, that whether your compound is thermolabile or thermosensitive. So you need to highlight this also. If you don't have thermostat fitted with your HPLC system, I think there is again a limitation in your nature of drug you are going on which you are going to develop a method. So because sometimes it happens when the resolution we are not getting any resolution, poor resolution is there. The increase in temperature improves the resolution. It's an experimental factor that sometimes the temperature at higher temperature, the resolution is much more and peaks are taller, and as per our expectation. So you need to look into that, whether your column is stored into a thermostat or not, whether you are able to control the temperature. If your drug is temperature sensitive, then obviously you need to look for a column which is thermostatically packed. So that is again an important factor. So these are the preferred experimental conditions where you can start with and just make a sample of your test concentration. Suppose 10 ppm is your test concentration of your dosage form. Just start it, and then you can start this trial run with this chromatographic conditions. So it's a basic diagram which need to show that basic silica molecules. On the left hand side, you see that they are silanol groups are there, OH groups are there, and if your compound is polar in nature, if polar functionalities are there, so it will definitely make a strong retention of the column, and strong interaction is there, hydrogen bonding interaction will be there. And if your compound is having some non-polar groups, so it's weakly polar, so it will not retain much. So what will happen, you have different compounds where there is no polar functionality. So in this case, you can easily understand that this compound will stay for a longer period of time. This will be moderately interacted, and this is not interacted at all with the surface. So what will happen? The first peak, you will see that non-polar compound is there, then the moderately polar, and then the highly polar. So this is a basic function. In case of reverse phase, this is a diagram of a normal phase mode where we are not changing any group on the silanols, on the silica particles. Where if we wish, and this is applicable for the separation of the polar compounds. And what need to do for the non-polar? Non-polar, we need to this silanol groups. The part of this silica particle is carbon loaded with C18. So this process is known as, if there are 18 carbon loaded, so we can say that it's a C18 column. If there are 8 carbon is loaded, we can say it's a C8 column. And C4 column, butyl, octyl, octadecyl. This way the nomenclature begins. So we are converting through this process. This is end capping also. We are converting a polar phase into a non-polar phase to separate the non-polar eluents. So this is the basic difference between a normal phase and a reverse phase. And this is the most used phase chromatography in pharmaceutical industry. So I wish to make you understand that since suppose we have taken a triazine, which is a herbicide, as a sample, and we wish to work on a concentration of 10 ppm. So what will happen? You will see the 4 diagrams, chromatograms. This is our computer simulated chromatograms, A, B, C, and D. So with the initial condition, as mentioned in the previous table, the only remaining decision before the first sample injection is the percentage of organic phase. I listed that 80 to 100%. So one approach is that let us start with the isocratic mobile phase of some average solvent strength. Like isocratic mobile phase means so 50% of A and 50% of B. 50% is buffer, 50% is organic. And you can see the diagram, chromatogram, that in the first one on the left hand side, this one, that only 3 compounds have been eluted. But the sample information says that it has 6 compounds. So initial triangle with the 50-50 isocratic phase, we are not able to elute the entire 6 compounds. However, the sample contains total 6 compounds. So what is the next step? That we can increase it to, for a better resolution, we can use a very strong mobile phase. Suppose 80 to 100% variation, and then reduces the organic phase accordingly, as per the diagrams, as per the illusion of the different peaks. So you can see that the 3 chromatograms have been illustrated in this slide, B, C, and D. These are the trial runs where the organic phase is modified from 80 to 100%. And the initial separation for the on the top right, that is B, it is 100% organic modifier valve run in this case. And you see that still we are not getting the chromatograph as per our range, but we are getting the peaks, all the 6 peaks after putting it 100%. It means the concentration of organic modifier need to be changed. So if we run with 100% organic phase, and we saw this response on the column. So this approach, the initial separation with 100% B results in the rapid dilution of entire sample, but only 2 band groups are separated. So we can now decrease it to the solvent side to the 80%. We showed a rapid separation of all the 6 bands. You can see in the D, and which is on the right hand side, the D chromatograph. You can see that there are 6 peaks, and there are the peaks are a little bit as per the system suitability, as per the expectation. So we can increase, we can further, we have, the scientist has decreased the strength to 60%, and which provide improved resolution, but a much longer run time with a broadening of later bands and reduced detection sensitivity. So what, I will show you another slide. In the run E, you can see that an alternative to initial isocratic separation is the use of gradient dilution. And you can see this E chromatogram. There are several, we run the gradient dilution, and in the gradient dilution, we changes the mobile phase from different concentration, from 80 to 100, 70 to 100%, and on different concentration, different compounds eluted. So since it is a multi-component analysis, as I told you, that isocratic separation may not be suffice the purpose of eluting all the compounds with desired system suitability parameters. So it's always better whenever you are going for the multi-component analysis, you can change, you can program your mobile phase. So in this elutions, we have programmed the mobile phase, and on different concentration, like 60% A and 40% B, or vice versa, 60% B and 40% A, different combination we can run in a single injection of 30, 40 or 1 hour in a gradient elution. So in 60 minutes, you started with 70-30, and you came back again to 70-30. So you, in between, you can change them or mobile phase concentration. So different peaks will have different retention factors. So they will definitely elute out. So you will say, once you have recorded this trial run, you can always go for the validation parameters also. So this is a common strategy how to develop a HPLC method with the initial conditions. And after seeing the response of this trial run, you can always change the mobile phase, or you can change the column itself, or you can change the mode of isocratic separation, from whether you are going to use isocratic or you are going to use gradient. So accordingly, you can use that. So now, after the initial run, you have the chromatogram, and you need to now improve the initial trial run. That is the optimization part. After the initial trial run, you need to optimize the conditions. The separation achieved in the first 1 or 2 runs usually will be less than the adequate, we can understand. And after a few additional trials, it may be tempting to accept a marginal separation, especially if no further improvement is observed. However, experienced worker, they realize a good separation require more than a minimal resolution of the individual sample bands, particularly for a routine procedure. So separation or resolution is a primary requirement for any qualitative or quantitative analysis. Usually a sample containing 5 or few compounds, baseline resolution should be, or resolution should be greater than 1.5. And resolution usually degrades during the life of the column, and can vary from days to days with minor fluctuations in separation conditions. Therefore, values of resolution is equal to 2 or greater than should be the goal during the method development for simple mixtures. And such resolution will favor both the improved assay precision and greater method ruggedness also. Sample containing 10 or more components will be more difficult to separate, and hence the separation goal often must be relaxed to resolution of 1.0 to 1.5. So that way we can always look into it. Narrow peak heights are always desirable. Gaussian peaks are there, so you know, and pressure of less than one bar, 150 bar is desirable, and means pressure between 2000 to 4000 psi is generally acceptable, but 2000 is ideal. If the sample run is showing a large amount of pressure, it means you need to look into the sample, whether any concern whether you can dilute the sample, or the column which you are using, whether what is the shelf life, or whether the proper cleaning is done. You need to do some troubleshooting. If you want to develop anything, you need to have the clear knowledge of troubleshooting. So whenever any technician from the Water, Saginet or Shimadzu is coming to your campus, just sit with them, just look into the troubleshooting, how they are doing troubleshooting. So because this machine needs a lot of troubleshooting, and analysts must know the troubleshooting. Otherwise, it will be difficult to time and again call the engineer and look for the possible solutions. So as an analyst, you must be trained. It's part of a developmental technique. You need to look into these parameters. So separation time, again, the run time is 5 to 10 minutes is desirable. I think if you are working on isocratic mode and if you are doing separation, it should be less than 10 minutes. So otherwise, unnecessary running the column with the mobile phase is a waste of time, and it will add cost to the company also, cost to the product also. So we need to look into this factor also. So there are other optimization chromatographic conditions which you can use. You can trial thing, and that you can choose columns who have long lifetimes at operating mobile phase. You can use bonded phase or high purity. You can use the C18 or C8 bonded phase first for good retention and resolution with typical acidic and basic neutral samples. So you can always start with low pH. We have seen that best peak shave is obtained when you are using it, working at a low pH, and pH 1 to 3 with the 20 to 50 micron. This buffer system, phosphate buffer system, is always giving the best peak. Avoid using buffer having sodium hydroxide, which is very much troublesome for your, or you can say it's a column killer. So you can adjust obviously the organic phase for maximum resolution and retentions. So you can, these days, you can always assist your development part with the computer assisted tools also, and runtime adjustments also. So I will skip this isocratic and gradient dilution. As I told you that what is the meaning of isocratic and gradient dilution. On the left hand side below diagram, you can see there are 10 compounds, and we run the isocratic dilutions. The peaks were overlapped, and when we run the gradient dilution, the peaks are separated well. You can see on the right hand side diagram that gradient profile, how the peaks 47-minute run is there, how the peaks have been separated of a 30 multi-component analysis. So these kinds of, if your sample is complex, such complexity need to be addressed through gradient dilution only. You cannot look for a isocratic dilution for such compounds. So mobile phase, I have told you that you can see the representative chromatograms, how the organic phase have been changed. Like in first case, we use 90% of the organic phase, and in case of second, 80%, 70%, and then 60%. You can say the performance characteristics have been improved by changing the mobile phase composition. And mobile phase means the organic modifier composition we have changed, and you have seen that good resolution of peaks are there. So once you have developed any method, you need to ensure that the method should be validated. Developing a method may be a you can say maybe a goal achieved, but the goal need to be validated. So ICH has published the guidelines for the validation of your method. You must do your validation. Otherwise, without validation, there will be of no use. Nobody is going to accept your research in terms of research paper or any thesis you have submitted anywhere, or you wish to add it as a monograph. You need to prove that your method is valid for the intended purpose, with the purpose you have developed the method. So this is the goal of validation. And in these days, how important the validation is. And generally, despite instrumental validation, despite the process validation, despite the method validation, these days generally industry do the human validation. Human validation means the analyst validation. They give you a certain test and they will check how you have produced the results. Suppose they have given you an essay to do it independently on the first day or second day of your joining in the industry, and you need to perform that certain test, and they have the parameter defined with them, the company, and they have the range defined with the company that if you have performed an essay and if you are giving a result of in the range of say 180 to 110, I think the analyst, the candidate has the potential to work as analyst. And if you are producing a result of 50% or more than say the desired limit, obviously you need to work out. You will be given training before straight away asking you for the sample analysis. So analyst validation is an important part. So everything these days need to be validated, whether it's a in silico techniques, you need to do it on validation test. There is a test set and there is a training set, you need to validate it. And if you are buying any goods for your home itself, like a ceiling fan, so obviously ceiling fan need to be validated. Company claims that this ceiling fan is running 200 RPM. So there are meters to check this RPM. You can check whether the company claims is right or not. So validation is an important parameter. The nutshell of this slide is that. So accordingly, ICS has defined that how to validate a developed method for HPLC. You need to perform the 9 tests. And these tests are very well defined in the ICH Q1, Q2 policy document. And you can perform these tests and you can tabulate the results of these tests, and you can look into the ranges described by the ICH for the same. If your results are coming in these ranges, then obviously your test is your developed method is validated and it will be accepted by the concerned authorities or for the publication of the patent purpose also. So accuracy, precision, specificity, selectivity, limit of reduction, limit of quantitation, and limit of linearity and range, robustness, and lastly the system suitability parameters. So these are to be validated. You need to carry out different tests. So I will just touch a bit of these tests, that why you need validation before introduction of a new method, whenever conditions are changed or any change in the method which is outside the scope of the existing method, you need to validate the method. So precision, generally you have seen that precision is done at 3 levels, that is repeatability, intermediate precision, which is also known as ruggedness and reproducibility of the results. So if you go through the definition of these tests, I think you will be not able to understand the proper concept, but because the definition itself is generic in nature, and you need to look into the experimental part of it. So I will discuss only the experimental part. Suppose you are working on the test concentration of 10 ppm, you just make 6 concentration of 6 dilution of 10 ppm and just take a reading of the HPLC area, and we need to calculate the mean, standard deviation, and the RSD. And the RSD should be less than 2%. If the RSD of 6 subsequent injection is less than 2%, it means your method is precise, and you can do it on intermediate precisions also with different equipment, different analysts, different days. And you can always reproduce the method on a different HPLC system at a different location by the different scientists. So it's a part, reproducibility is the part of transfer of methods. Suppose the Sun Pharma has a R&D center at Gurugram, and their team has developed a method, HPLC method, and they need to transfer this method to the Gujarat or the other branches which are doing some routine analysis. They are not involved in the R&D. So they transfer this method. Once the method is developed at the R&D center, they transfer this method to those locations. So they are producing the reproducible results which have been produced at the R&D center. So if the results are between the desired limits, then obviously you can say your method is reproducible. You can use this method at any location, on any machine, and anybody can use this method. So reproducibility is a part of technology transfer in terms of industry. So sometimes FDA inspects the industry and they ask the foreign locations of that particular industry to produce those results, which have been showcased by the drug regulatory department, drug regulatory affair department at the industry. So the analyst at the foreign location, they need to produce the same results. So it's an important exercise in industry. Linearity, obviously, you need to make the 6 concentration, and you need to get the area of 2 readings of each concentration, and you need to put into the graphs, and you need to check the correlation coefficient. If the correlation coefficient is more than 0.9999, it means your method is linear over a defined range. So you need to define the range also, in which range your method is going to work with the sensitivity and the specificity also. So you need to define the range, and you need to define the test concentration also. So minimum 6 concentrations are needed, and duplicate sample of each concentration, and you need to calculate the average of those duplicate concentration, and then you can always calculate the correlation coefficient. So this is very much needed to define the range. Robustness, obviously, sometimes it happens that the analyst have not used the chromatography condition very well, or some interns have feed the data, and there are small variation have been done. But so is your method is robust enough that although the deviations are small, but results outcome should not be very much significant. So whether your method is robust or not, if we do small but deliberate variation in the method parameters, you can use some mobile phase within plus minus 2%. So the RSD for such analysis should not be more than 2%. So that is the purpose, that erroneously sometimes it happens that the flow rate was 1.5 and you have put flow rate of 1.35. So how much it affects the parameters. So you need to check this robustness. It's a small deliberate variations. So another factor is limit of detection, which is limit of detection is the smallest amount of concentration of analyte that can be detected only, but need not to be necessarily quantitated. Means you how below your method, how far your method can reach to which concentration. But at this concentration, we need only detection, and we need to identify the limit of quantitation, which is the lowest level of analysis that can be quantitated to some degree of certainly a precision. Means the difference is that you need, if you are saying that 10 ppm or 5 ppm is your LOQ, the 5 ppm LOQ is there, so you need to have 5 readings of the same concentration, and your RSD must be below the 5%, or your signal to noise ratio should be comparable at the level of 10. So LOQ is equal to 3 times of LOD. So that is the main purpose. So at LOD level, you say that 1 ppm is our LOD, but you need not to run the 6 samples, there is no concept of RSD, percentage RSD. So you need to calculate the LO. In case of LO, you need to calculate the precision again, and it should be within the range of say 5 to 10% as per the protocol. So this need to be identified and validated. This is needed because this will give you an idea of sensitivity of your method. And these days, the signal to noise ratio is also used as a mathematical tool to calculate this LOD and LOQ, and you can always validate this within an experimental condition also. So both way you can do that. You can use the data of your chromatograph, and you can use the software defined LOD and LOQ. Lastly, the system suitability parameter is of greater importance, and every software generally gives this system suitability parameters. Inbuilt softwares are there. You need to check the system suitability parameter for any run. Any run you are doing, you can get these data. So you must look into the tally factor. That should be less than 2. Resolution should be greater than 2. If multi-component analysis are there, then you can always have a fair amount of liberty of 1.5 also. Injection precision, it again defined by the industry to industry and the most of the pharmacopoeia, but it should be less than 2% in most of the cases. Theoretical periods obviously is an important factor, that it should be more than 2000, and these are the mathematical factors which can be calculated very well. And capacity factor defines the distance between the 2 peaks, the separation between 2 peaks, how 2 peaks are separated very well. So these are the system suitability parameter. After doing all your development part, please do take a printout of all the system suitability parameter and check with the ideal range whether your analysis is following the ideal ranges or not as a part of system suitability. So I think this is all. I have to more than I think the desired time which is allocated to me. Sorry for that on a Sunday, but I am open to any queries. If you want, please.
Thank you so much, sir. Thank you so much, sir.
Thank you, Ji. Thank you.
Sir, we are very grateful for the times and efforts you took to share your thoughts and experience with us. Sir, your presentation is so much informative and sir, very interesting, and sir, so much learning experience we have gaining from your presentation, sir. Sir, your lecture was very inspiring for all of us. Sir, one question from participant side. Sir, how can we improve stability and solubility of component?
Stability and solubility of component, I think it's beyond the scope of this discussion. We are doing for HPLC method development. Okay. Although we can look for the stability indicating HPLC method, obviously stability it cannot be achieved by the chromatographic conditions. You need to do some synthetic or some polymerization with the drug. But in case of chromatography, obviously you need to look into the stability of the sample itself for suppose 12 hours or 16 hours or 24 hours. You need to keep the sample for 24 hours and analyze the sample again on the next day, whether you are getting the same response. If you are not getting the same response, it means the stability need to be addressed, and you need to mention in your development of the chromatogram that the sample should be freshly prepared, or the analysis should be done within the 3 hours times. So this we have so many drugs which are not stable, and immediately after opening the vial, you need to do the analysis, and after 1 hour you are not able to do the analysis because of the raise of the impurity level.
Thank you so much, sir. Very well explained, sir. Sir, next question is, how can we reduce fronting of chromatogram?
Which one? Come again, please.
Sir, how can we reduce fronting of chromatogram?
So I think these days the softwares are there. They do these kind of the adjustments very well. And if you are using an old software, I think I don't know, but the Empower software and Agilent Leeson software, they generally improve your chromatograms. That is not an issue with your check your column properties also. these are the two factors which can help you.
Thank you so much, sir. Now I would like to invite Dr. Ashwini Dhingra, HOD of the department, for vote of thanks.
Thank you, sir, for your valuable session.
Thank you.
So good afternoon, all of you. On the behalf of Guru Gobind Singh College of Pharmacy, with due respect, I would like to extend my heartfelt thanks to our worthy speaker for sharing his valuable knowledge and experience with us. I appreciate your knowledge and experience for conducting the research in the design and development of pharmaceutical. Your expert talk has provided a brief overview about liquid chromatography, various techniques while selecting a particular analysis. Apart from this, your kind presence and expert talk in this program also encouraging us, and we are also hoping good health and future cooperation with us. Thank you, sir.
Always
Thank
to
Thank you.
look for my students. So many students are there. Ashwini Dhingra is my first batch student, and how our students are excelling these days, I think it's a real, you can say, it's a real outcome of the education system, where you are working with your students, and your students are motivating you at this age. I'm motivated by Dr. Prerna, Dr. Dhingra, Dr. Bhamna, and so many students. Other students are also listed in the participant. So you all people give the motivation us to keep our knowledge updated.
Sir, you are the role model for us.
Sir, you were also motivating to us also, sir. Sir, I remember also on first day of my job, you are also inspiring to me also, sir, what we have gaining and what's the scope and what's my aim. Sir, you are also inspiring to all of us.
Yes. So if any whomsoever has raised the issue of fronting or tailing, they can write to me also if they are facing a particular problem with a particular component also. I advise them to check the column properties first, because these are the column parameters which need to be checked. Otherwise, it can be an integration issue at the computerized level also. So my mail ID, I think I showed in the first slide, and you can always write to me, and I love to answer these queries.
Thank you so much, sir. Sir, now as a token of respect and appreciation on behalf of Guru Gobind Singh College of Pharmacy, I would present a certificate. It's on the screen.
Thank you. Thank you, Ji. Very well received. Thanks for giving me a platform to exchange my knowledge after such a time. And I love to interact time and again with the young minds, and it will give me a feeling of being young rather than looking an old fellow. So once again, congratulations for organizing such platforms.
Thank you, sir.
Thanks a lot, sir.
Thank you, sir.
Thanks to all the participants for joining today's session. We will again joining at 2 and for third technical session. Participants, you can join again 2 p.m. around for third technical session.